So thank you all for coming today. I'm going to be talking about early life stress and its correlation with BDNF methylation and major depressive disorder. I just want to start off by going over what I'm going to be talking about today. Um, Overall, I'm going to be talking about how childhood trauma correlates with gene methylation and therefore influences a risk of symptoms for major depressive disorder. Gene methylation alters the gene-related brain function, which then alters behavioral aspects, which is how this whole process is going to work. And overall, in my research, I investigated the correlation between early life stress, major depressive disorder, and brain-derived neurotrophic factor methylation. I just want to define a few terms before I get started with my discussion. Firstly, brain-derived neurotrophic factor, or BDNF, which is the aspect of DNA, which is a gene which is responsible for emotional regulation and neuroplasticity. And then major depressive disorder, or MDD, which I will be referring to it as later in the presentation, is a serious mental condition that includes symptoms of extreme sadness, loneliness, loss of interest in activities, loss of appetite, loss of sleep, and suicidal thoughts or behaviors. Early life stress is a broad umbrella term that covers all aspects of early life trauma and childhood adversity. This can range from childhood abuse and maltreatment to war and natural disaster and many things in between as well. Next, epigenetics, which is the regulation of gene functionality. It can kind of be thought of as like a dimmer switch on a light switch when it's all the way, it can either be all the way up and have full functionality of the gene, or it can be dimmed and have the gene not be functioning as much. DNA methylation is a process of is an epigenetic mechanism in which certain aspects of DNA are being altered slightly. And lastly, I want to talk about the hippocampus, which is a region of the brain that is responsible for memory retention and emotional regulation. This is the region of the brain that I found was most relevant in all of my research. The methods I used in my research consisted of a database search using Google Scholar and the PubMed database, using the keywords epigenetics, depression, adolescence, transgenerational, BDNF, DNA methylation, trauma, and early life stress. I excluded several articles, and I excluded them if they were written before the year 2019. They were a literature review or another type of review that was not a study, and they did not mention the specific terms of DNA methylation, BDNF expression, and childhood trauma or early life stress. I varied the outcomes of my articles to show a wide array of possibilities, as well as using both human and animal models and a range of participant age. I'm gonna now transition into my discussion, with my first theme being early life stress and epigenetic mechanisms of BDNF methylation. I'm gonna be talking about how the early life stress and childhood adversity directly influences the methylation of the gene. The first study that I looked at focused on BDNF methylation in individuals who were diagnosed with PTSD as a result of childhood adversity and early life stress. The study consisted of 180 participants, 99 of which had a PTSD diagnosis. The researchers conducted a genotyping study in which BDNF methylation was measured via DNA extraction in the blood, and they compared the genotyping in the PTSD participants and the control participants. They found that there was not a significant enough correlation in comparing the control participants and the PTSD participants. However, there was a significant correlation when the presence of the GLE of OXTR was studied in the PTSD patients. And the presence of this gene indicated BDNF methylation. Additionally, not looking at the PTSD patients, but looking at the control participants, the control participants who had reported childhood adversity also experienced BDNF methylation. This was significant because there was no BDNF methylation in the participants with PTSD unless the gene was present. However, there was BDNF methylation in the control patients who reported early life adversities. This led the researchers to conclude that the BDNF methylation is not necessarily a direct result of a PTSD diagnosis, but rather is a biomarker for risk of major depressive disorder symptoms as a result of childhood adversity. The next study that I focused on looked into the impact on BDNF levels following childhood trauma. This study consisted of 190 young adults ranging from ages 10 to 19 who had no previous history of psychiatric disorders. They took the childhood trauma questionnaire, which has a range of 5 to 125 to 125, broken up into 5 subgroups ranging from 5 to 25. And this questionnaire has been used across several of the studies that I will be talking about today. They also took the perceived stress scale, and the participants were measured for prevalence of the Val66 met polymorphism, which is when in the DNA, the valine is replaced by a methionine, which is a polymorphism and a modification of the BDNF gene. And additionally, hair cortisol concentration levels were measured, and the results were compared. There was not a correlation between the hair cortisol concentration and the childhood trauma questionnaire scores, or between the Val66 met prevalence and the hair cortisol concentration levels. So neither of these relationships on their own provided a significant correlation. However, there was a 3-way correlation between the hair cortisol concentration, childhood trauma questionnaire scores, which were higher scores, and the Val66 met prevalence. However, this relationship was only found in female participants. This 3-way correlation showed that the Val66 met prevalence provides an increased risk for major depressive disorder symptoms. Additionally, since it was only prevalent in females, it showed that women may be more at risk for major depressive disorder because they experience a higher difference in HPA axis signaling, which is an indicator of early life stress. This graph taken from the study compares the male and female Val66 met individuals who had the Val66 met polymorphism, comparing their hair cortisol concentrations with their childhood trauma questionnaire scores. And you could see the significance of the p-value, which demonstrates significance in the male graph is shown to be insignificant, but it's significant in the female graph on the bottom. This is just looking a little bit closer at this graph, just so you could see it a little bit there. The third study that I looked at was comparing the lasting effect in this theme, was comparing the lasting effects of BDNF methylation in response to early life stress. So this study looked at men and women who were over 65 years old, living in the Esprit cohort in France, which was a mental health study. Participants took baseline data and a trauma survey to assess 3 kinds of early life stresses: poverty, childhood maltreatment, and war and natural disaster. And BDNF methylation was measured at promoter regions 1 and 4. The results showed that there was only significant methylation in participants who experienced war or natural disaster. The participants who experienced childhood maltreatment and poverty did not have significant BDNF methylation. So the researchers could not conclude definitively that there was a correlation between ELS and BDNF. However, this also introduced the possibility that since it was such a long time between the experience of the ELS and the study being taken, that there was a possibility that BDNF methylation can reverse itself over time, either partially or fully. Additionally, since war and natural disaster was the most severe ELS that was studied, and it was the only one that showed up to be long-lasting methylation, it was also concluded that the more severe the early life stress, the more intense the methylation. And this is comparing two of the graphs that were taken from the study. On the left, comparing the financial difficulties and poverty ELS with the war and natural disaster ELS, and you can see there's more difference, a bigger difference in the graphs on this side than there is on this side. To sum up these three articles, I want to emphasize that there is, in fact, a correlation between ELS and BDNF methylation. However, over time, this correlation was not seen as well because the process reverses. Additionally, BDNF methylation intensity is correlated with early life stress severity. My second theme of my discussion is the role of BDNF and ELS in brain function and depression. The first study that I looked at was how BDNF in adolescents with trauma-related depression was methylated. This study had 48 participants, 30 of whom had a major depressive disorder diagnosis. All participants took the childhood trauma questionnaire, which was seen in previous articles as well, and took a depression inventory as a baseline. Researchers measured BDNF protein levels and PMTOR levels, which regulate neural growth and protein synthesis and are very closely correlated with BDNF. The results determined that with an increase in major depressive disorder, there were significantly lower BDNF levels. Additionally, in participants who experienced suicidal behaviors, they experienced BDNF lowers that were even lower than the major depressive disorder participants who did not have suicidal behaviors. And this is demonstrated on the two graphs below, in which the darker green color has significantly lower serum levels here and here, with the suicide or self-injury graph showing a larger difference in serum levels. The next study that I'm going to be talking about is how the Val66 MET polymorphism correlates with major depressive disorder. This is a polymorphism that I touched on before, which it's a variation of BDNF. This study focused on a very specific group of 20 to 30-year-old Chinese men with no previous psychosis diagnosis. And researchers took MRI brain scans of brain activity and additionally genotyped for the Val66 MET polymorphism via DNA extraction and blood sampling. They found that with a higher childhood trauma questionnaire score, there was an increase in the prevalence of the Val66 MET polymorphism and additionally a more severe major depressive disorder. And the higher the childhood trauma questionnaire scores were, the higher the major depressive disorder scores were, showing a correlation between more severe childhood trauma and more severe depressive symptoms. Additionally, the prevalence of the Val66 MET polymorphism correlated with a lower BDNF expression, showing that as there's an increased prevalence of this variation, BDNF expression is severely regulated and BDNF proteins are not produced as much. And finally, a higher childhood trauma questionnaire is correlated with lower neuronal functioning, which means that the brain was not functioning as well in response to the childhood adversity. The significance of these results is that the severity of the childhood adversity was directly correlated with the intensity of the major depressive disorder, which is also directly correlated with the prevalence of the Val66 MET polymorphism. In response to the early life stress, the BDNF levels were suppressed, which decreased neuroplasticity, which is something that BDNF is responsible for, as well as emotional regulation, which led to the increase in major depressive disorder symptoms. Here are images that were taken from the study of the MRI brain scans, showing different areas of the brain being highlighted in response to different different kinds of stimulation. This kind of represents the neural functioning that I was talking about was being impacted. Next, I'm going to talk about the study focusing on the indirect effects of ELS on major depressive disorder through the BDNF levels. This study hypothesized that these things weren't all correlated together, but rather the BDNF was connected to both ELS and MDD, but they were not directly correlated to each other. This study consisted of 64 participants who had a major depressive disorder diagnosis and 70 control participants. All participants took the childhood trauma questionnaire, which focused on sexual, physical, and emotional maltreatment. And methylation was measured at promoter regions 1 and 4. The results showed that there was a positive correlation between higher childhood trauma questionnaire scores and BDNF methylation. And since there was an increase in BDNF methylation, neurocognitive abilities were decreased. However, there was an inconsistency with this and some of the other literature that I reviewed, because not all of the major depressive disorder patients had significant BDNF methylation. This can be attributed to what the study hypothesized, that not all major depressive disorder patients experienced early life stresses and had the direct correlation of the early life stress on their depression diagnosis, therefore not having BDNF be as significant of a factor. This led to the discovery that not all major depressive disorder patients are going to be uniform, as there are a wide variety of factors that can influence major depressive disorder. To sum up these past 3 articles that I talked about in this theme, there is a correlation between early life stress, BDNF methylation, and major depressive disorder, but it is hard to measure this because there are so many factors contributing to a major depressive disorder diagnosis. And additionally found that BDNF is a biomarker for MDD risk rather than a direct cause. My third and final theme that I'm going to be talking about today is treatment for major depressive disorder, specifically targeting BDNF methylation in the context of early life stress. In the next 3 studies that I'm going to be focusing on, all of them used a rodent model because they were all testing prospective treatments for MDD and could not use a clinical human model. The first study that I'm going to be talking about is studying the epiglocation 3 gallate, or EGCG, as I'll be referring to it from now on, in a rodent model. This study utilized 4 groups of mice, the first group being healthy with no presented adversity and no treatment. The second one having experienced the CUMS protocol, which is chronic unpredictable mild stress. This mimics early life stress as it would be experienced in humans, causing depressive-like and anxiety-like symptoms. The first group of CUMS mice were not given treatment. The second group of CUMS mice were given the EGCG treatment, and the third group of CUMS mice were given clomipramine, which is a clinically approved and FDA-approved antidepressant. The researchers measured brain inflammation and structure by extracting pieces of the rodent brains after treatment and behavioral testing, as well as BDNF expression. So the results showed that the mice who underwent the CUMS protocol had significantly decreased BDNF expression, as well as a damaged hippocampal region in the brain, which was found to be significantly inflamed, and the structure was modified. And they had a significant increase in immobility as well. In both the EGCG treatment and the clomipramine treatment, The BDNF levels were partially restored, and the hippocampal damage was partially repaired. And both treatments saw a decrease in immobility. However, the EGCG treatment had a higher decrease in immobility than the clomipramine treatment did. The significance of this is that there are similar or improved qualities in the EGCG treatment when compared to the clomipramine treatment. Therefore showing that EGCG could be potentially a very useful treatment for major depressive disorder, but it requires further study in a human model in a clinical setting. The image that you see on the left is comparing the 4 groups of mice and specific types of cells and differences across the kinds of cells. The next study that I'm going to focus on is studying the jujubicide A, or JUA, as it could be used as an antidepressant. This study also used a rodent model, and instead of using the COMS protocol, it used the colorectal distension model, which also had a similar effect of inducing the same effects of early life stresses. However, instead of humans, it's in mice, and also inducing depressive-like and anxiety-like behaviors. There were 2 groups of the mice, a group that got a daily dosage of the JUA treatment for a span of 2 weeks, and a control group who was given a saline solution for the same amount of time. The results showed that the CRD protocol reduced BDNF expression significantly, which was seen similarly in the COMS protocol, as it reduced BDNF expression, caused significant neuroinflammation, and increased depressive-like and anxiety-like behaviors. The rats who were given the JUA treatment had significantly lower major depressive disorder symptoms than the control group did. And the JUA was seen to restore BDNF levels and reduce the neuroinflammation. This also shows that, like the previous study, the JUA is effective in being an antidepressant in the rodent model and would require further study to be tested as an antidepressant in the human model. Finally, I will be talking about the study that investigated the effects on intranasal insulin on PTSD-induced rats. It is important to note that these rats were induced with a specific type of depressive-causing disorder. Post-traumatic stress disorder has been known to cause depressive symptoms, and it is as a result of an experienced trauma, but it is important to note the difference. The methods of this study also had 4 groups of mice, a control group who received no stress protocol and no treatment, a group that experienced the single prolonged stress protocol but were not given treatment, a group that were given the single prolonged stress protocol and were administered intranasal insulin, and a group that was only administered intranasal insulin. Intranasal insulin is administered through the nose in order to bypass the blood blockage that would in order to have a faster pathway to the brain instead of being administered through the blood. The researchers measured the behavior of the mice as well as BDNF protein expression. The study showed that the SPS protocol increased depressive-like symptoms, which is not surprising, as the stress-inducing protocols also in the previous 2 studies also had the same influence. And additionally, the SPS protocol decreased BDNF expression and increased neuroinflammation. And the intranasal insulin treatment partially restored the BDNF expression and reduced neuroinflammation in the mice. At the bottom of my slide, you could see the process in which this study was carried out. The initial SPS treatment was given at the start of the study. There was a 14-day period in which intranasal insulin was administered, and then several days following in which behavioral tests were conducted before the animals were used for brain studies and genotyping. The significance of the results of the study was that intranasal insulin was seen as an effective antidepressant in the rodent model. And like the other two treatments that I looked at, it could have the potential to be translated into a human model for a clinical purpose, but it would require further study. To summarize the past three articles in which I talked about treatment, all three of the antidepressants that were studied were effective because they reduced the BDNF methylation and decreased neuroinflammation, as well as reducing depressive-like and anxiety-like behaviors in the rodents. All three showed a potential for translation into a human model upon further study. To wrap up my presentation today, I wanted to give a broad and defining statement that there is a positive correlation between early life stress, brain-derived neurotrophic factor methylation, and major depressive disorder. There were variations across the articles due to age and time after the stress and a variety of depressive factors, in addition to severity of early life stress. There are several gaps in the research that I wanted to address. And two of the major ones were challenges in measuring major depressive disorder in adolescence, because there is so much happening biologically in adolescence with development. It is hard to measure directly the effects of ELS and how it correlates with major depressive disorder. And additionally, on that note, addressing direct causes of major depressive disorder is challenging due to the wide range of causes. To address these gaps, a more holistic approach should be taken in addressing major depressive disorder, both genetically and with a questionnaire, specifically with the adolescents, having a wide variety of questions on a questionnaire that could address the many types of factors going into what could be causing the depression would be extremely helpful in having more studies about how the adolescent brain is functioning, and as well as having further studies in the genetics of major depressive disorder in order to further define how the disorder works and get better treatment studies so that the condition can be improved. Thank you so much for listening to my presentation, and I would love to have any questions.